So, good afternoon again, and welcome to the second session of this afternoon. We begin by a talk from Florian Hollfelder from Cambridge, who uses directed evolution to evolve new chemicals.
Thank you, Clemens. When I was an undergraduate, I was interested in mechanism and was led to believe, because there were so many structures coming out at the time, that it would be a piece of cake to make functional proteins that would be better than anything that chemical entities and enzyme models could do. And that was definitely a bio-inspired statement by my teachers at the time, but protein engineering and protein design has been more difficult than the expression engineering that kind of puts you on the level of a plumber promised to be. It's more complicated than that. And I guess it's fair to say that we don't really understand how proteins work if we apply the Feynman test. I do not understand what I cannot build. And so I'd like to broaden, and that's already shone through a bit in the morning, the definition of bioinspiration by not just being inspired by what we see, but also by the processes that nature must have employed to bring about the enormous diversity and the precision and the power of functional molecules that we see. So, so the process, of course, is Darwinian evolution, and no one will be surprised about to see cycles of testing fitness, of amplification, of neurogenesis, and so on, that shape evolution in some way. But I want to talk about mainly this aspect here, the screening aspect. Is there a system that we can use in the laboratory where we are as rigorous or as tolerant as nature, the eternal tinkerer, is in providing thresholds to find out fitness differences and deal with DNA libraries, either man-made or from natural sources? And so you see about 5 of these cycles, and the question is whether there are technologies that make it relatively easy to work with such cycles, even though these processes are open-ended, and they're almost the opposite that a young physical organic chemist would expect, because they're to some extent random. Is there still something coming out that's useful and understandable by the end of the day? And one problem there is that we're facing an enormous challenge by numbers. If you take a protein that's not even the smallest one with about 300 residues, each of those amino acids could be replaced by 19 others, and that can happen in all sorts of combinations throughout the protein. So the sequence space that you'd have to cover to be comprehensive and test all possibilities by far exceeds the number of atoms and molecules in the universe that we've already seen. And so even if we would just focus all our divine energy onto evolving one protein, then there must be shortcuts and there must be other ways of doing it. But the available gene repertoires that we employ reach up to 10 to the 15. Most of what I talk about is actually smaller libraries, but that's a concentration of sort of DNA in maybe 10 mils, the maximum experiment that you have in a molecular biology lab. So our repertoires are much smaller than that number, and even smaller in comparison to the available sequence space for proteins. So at least we want to do well on getting somewhere in that direction and screening very efficiently. So we can never do enough, but at least we want to go beyond, again, what you can do with your 2 hands and 2 legs in the lab. Okay, now, if we want to learn about the process of evolution, then of course the cell plays an important role. And the reason why it does that is because once you make a library, that would mean that you have a large number of different DNA molecules that then encodes for a protein. It doesn't help you if you just identify the protein. The protein cannot be decoded as such, but if you have a genotype and phenotype linkage, then you can keep protein and DNA together. And of course, the cell is a very complex genotype-phenotype linkage, and maybe for engineering purposes, too complex. So wouldn't it be nicer if we could evolve just one thing at a time? We can maybe talk in the discussion afterwards about what it means to evolve in vivo. That can be done. But to be stringent and to exploit the numbers just for the one protein that you're interested in, for example, that may mean a bit of reductionism. And that reductionist model is seen here. That is for at least the next 20 minutes, doing everything that a cell normally does. We make water and oil emulsion droplets, slightly different to the bilayered structure we've seen before. There's just oil on the outside, water, and a small interface, no bilayer over here. And then we put into those water and oil emulsion droplets that are stabilized by a surfactant, an identifier, that would be the DNA, and that concept can be extended to maybe even encoding small molecules, and we co-compartmentalize it with the functional molecule that's made from it. In the simplest case, you have DNA and then RNA or protein by in vitro transcription translation, and we'd like to read out whether this clone is a good one or a bad one. Okay, so protein and DNA belong together, and the signal, the real phenotype, is, for example, the product of an enzymatic reaction that's also enclosed in here. Now, we've been working on this for maybe 10 years, and unfortunately the referees had to point out that these concepts are not entirely new, because in '54, Joshua Lederberg already showed that the one cell, one antibody rule by using such compartments. So the history of this as cell mimicry is relatively old. Rothman has used this to Poisson distribute single enzyme molecules and did one of the first studies of single enzyme molecules. And then that technology was reawakened without them actually knowing about this historic work by Andrew Griffiths and Dan Torvik, who treated it as an evolutionary unit and for the first time performed in vitro evolution with these in vitro compartments. Okay, now we have to sort of put them into perspective. They are about as big as a cell. The diameters are typically between 2 and 50 micrometers, so slightly larger than cells, but are very small still and have pico to nanoliter volumes. And we treat it effectively as an ultra-small test tube, where if you use expensive reagent or very dear reagents, you do that with sort of a minimal volume that you have to fill, and therefore you can do things at a throughput of millions or more that normally would be far too expensive to ever contemplate. The simplest way of droplet formation is one that just involves a stirrer, an emulsifier, and allows you to make, by just a few minutes of emulsification, to create about 10 to the 10 of these compartments that in 1 milliliter of this kind of milky paste that comes out, and that in the cartoon looks like that. And that's already equivalent to 1 million of relatively high density multi-well plates. So even with just this small trick, you get into very high throughput already. And that's been used for direct evolution by a number of people, most notably Phil Holliger from the LMB, who's evolved polymerases very successfully. And then there are a number of other people who have also used that principle. And I just want to illustrate what we have here, an ability by an in vitro compartment to create a genotype and phenotype by an experiment that makes this genotype-phenotype permanent. And that's one where we take a piece of DNA that has a small molecule attached to it that's here shown bigger than the DNA, but of course in reality it isn't. And then we add the kind of in vitro expression kit that we've heard about in the morning, the pure kit or another ribosomal extract system, and make protein from it. And then a protein is made that is abbreviated as a protein of interest, and it has a little tag. And that tag is an enzyme that can react with this substrate over here. So you express a protein, and then this end bites back to label the very DNA that it's been made of. And as a consequence, you then have, if you do this in a droplet, a situation where you've made maybe 10 to the 9, 10 to the 10 different droplets, compartmentalized DNA into each of those. Maybe you don't fill all of them to make sure that per droplet you only have one according to Poisson distribution. And then each of these DNAs is automatically, because you have the compartment, labeled by its own DNA. And then you can start doing a selection cycle. And this is what I show here, the first Darwinian selection cycle. Here's your IVDT mix and the DNA that's labeled with the substrate. You form your emulsions and then create this display construct that has a protein and the DNA that contains all the information for it attached to it. And then you go through a round of what's called affinity panning, where you pull out, in this case, protein binders. So, for example, you could make antibodies or alternatives to antibodies in this way. And this was just to illustrate how we can use the compartmentalization for making an in vitro permanent genotype-phenotype linkage. What I show here are the alternatives to that technology. Here, for example, is one that's used to make most of the antibodies that are used to therapeutics, for example. It's phage display, where you employ E. coli to assemble a phage, and then the DNA inside of that phage construct is displayed on the outside and can be used just in the same way before as a display construct. Now, here is a system that's called ribosome display. It achieves the same thing, but in an in vitro way by fixing the RNA that sort of goes into the ribosome, and then you stop by encoding the ribosome as it's about to leave the RNA, change the magnesium concentration, and fix that ternary complex. So again, you have genetic information linked to the protein, and that's a system that Andreas Plüchtun, for example, has pioneered. There's a similar thing called puromycin display devised by Jack Szostak, and so there's quite a number of these alternative display systems now, and the objective here would be to use it in vitro. These are maybe hard to use because they are made up of RNA, which degrades relatively easily. If you want to do chemistry, you maybe have to go to these systems here that employ DNA, and then you could put in not only alternative amino acids, but you could also do chemistry on these constructs because our DNA is so much more stable than that. And so since we're talking about biomimicry, that can be carried forth by employing the principle of multivalency. Here, for example, you have systems, and this is yeast display, a similar display construct, in this case pioneered by Dan Wittrup, where you can put multiple copies of the protein onto a yeast cell that contains the DNA. So you're still selecting in vitro, but you need to go through the organism, which may constrain your libraries, but you can do a selection on top of it. And the cool thing about this is that you can count the number of fluorescent target molecules that are sitting on the molecule. So you can do something that looks a bit more like a real KD titration. And we've done effectively the same thing that we did to make this display construct, but had a preamble where we co-compartmentalized DNA with a bead and did a PCR reaction, and you get multiple copies, about 10 to the 5. And again, just by using the in vitro expression kit, you can assemble 10 to the 5 identical protein molecules with the DNA that encoded it. And we're now using that for selections to see whether when you have to go through a bottleneck of yeast, or when you don't have to go through a bottleneck, you get other selection trajectories. So that's convenient and relatively easy to make, but there are problems with bulk droplets because they look not like the cartoon, but rather like this. They're really very different in size, and if they're different by a factor of maybe 10 to 100 in terms of distance, you can imagine that in terms of volume, they'll be more different. So what do you do if you want to not select for binding phenotype, but for catalysis, where you would like to measure a concentration for a product? And the reason, and that's the reason why making monodispersed and homogeneous droplets became important, and they're made by microfluidics and enable you to do quantitative assays. These are typical pictures where everything looks the same, and then you can also put them into a waiting line, and you can do very carefully controlled incubations, and you can see how a rate really develops there. So how can that be done? This is work by Steve Quake, Francis Arnold, and also Dave Weitz, who formed droplets in what is called a break-off flow, where you have an aqueous sample coming from here. That would be your DNA solution, for example, maybe together with a substrate or with the in vitro expression kit. And droplets here are broken off by oil streams that come from the site and then make surprisingly monodispersed droplets at a high rate. So the rates here are 10 to the 3 droplets per second and 10 to the 8 droplets per day. So still respectable numbers, a bit less than the emulsification procedure I showed before, but still very fast and very good in terms of quantitative assays. And the technology then to turn this into something that a molecular biologist or a chemist can use is the soft lithography approach pioneered by George Whitesides, which makes it very, very easy to learn maybe in a couple of months how to make your own microfluidic chips and bypass hard design, but you can just try out which microfluidic systems and designs work for you. And then you need the microfluidic system that you've designed in here, and then just punch in with biopsy punches supplies to pumps and let a laser or something else interrogate whether you have formed product. Okay, so now we come to these catalytic selections, where we want to look whether we can do selections directly for multiple turnover, whether we can count the molecules that are made by a particular protein in a quantitative way. And for that to be successful, we had to sort of test that you can do biochemistry and biology in these droplets. And indeed you can. What you've seen just in the slide before was a PCR machine effectively, so you can amplify DNA, you can in vitro express, you can grow cells. In this case there were yeast cells. Here there are worms that we can grow in there, and you can do reactions. And if you're lucky enough and you have a product that doesn't leak, then this assay will be quantitative. And then a large number of people have developed modules for making droplets, for fusing droplets together, for sorting them, separating bright fluorescent ones from others. You can incubate them for long periods. You can split them, you can dilute them, you can take them offline into a syringe or a test tube. You can mix them by a little wiggle line and re-inject them and so forth. So that in effect you can design relatively complex workflows that allow you to dispense with pipetting and let a system that you've designed on your chip do much of the work. Now, these are designs that were made and implemented, and I'll just talk about one of them. It's probably still better not to be too complex, because remember you have to do all the same things for 10 to the 8 droplets, and a video that I show you also in here doesn't fully represent whether something works for hundreds of millions of droplets in the same way. But at least you have the ability now to design a workflow where addition of reagents, temperature shocks, and so on can be programmed into it. And I show you how we applied this to an evolution question. And the direct evolution experiment here was that we wanted to repurpose a promiscuous enzyme, an enzyme that already has multiple chemically distinct activities. This enzyme is called phosphonate monoester hydrolase. And the specific identity doesn't matter, but we used it as a springboard for natural evolution, because in order to traverse functional space in the most efficient way, it would be advantageous if you have an enzyme that already has even a low activity of the new function that it should maintain. And effectively what we're doing there is to try to find out whether enzymes already have the potential of low activities that then can be used for repurposing an enzyme catalyst by utilizing whatever is available in the active site, the reactive groups, nucleophiles, cationic groups, anionic groups, general acids and bases. And again, we did that in a superfamily that we and others have defined as being particularly promiscuous. So it's called the alkaline phosphatase superfamily after its name-giving member. And these are others that share the activities that are elsewhere in the superfamily. They do that, but they also hydrolyze other substrates that have never been seen in nature. And we come back to that theme later. But we just wanted to focus on one of these evolutionary shortcuts and evolve sulfatase into a phosphonate hydrolase. And this is the workflow that we then developed. We take E. coli that had already previously expressed the enzyme of interest here, and then upon droplet formation, we lyse it so that the enzyme is liberated and co-compartmentalize it with a substrate. Now, when you incubate that, you generate product eventually. These are very bad enzymes, so we have to work or wait for 4 or 5 days until this happens. So we put them into a syringe for the time being. And then we do a test, and that's a sorting experiment here that I illustrate with a movie later, and we go through rounds of DNA recovery, randomization, retransformation into E. coli, and again do a cycle of lysis and exposing them to the substrate to see whether they turn over successfully. Now, this will only be successful if we can measure kinetics here effectively, and these are typical data that we get. You see that you can reflect a better enzyme and distinguish it from a not-so-good enzyme very easily, but there is a bit of an error, and that error was, since we were neophytes and using microfluidics first, we thought we're just not detecting well enough. But in fact, that wasn't the problem. The problem is now that you're having phenotypic variation between single cells. So when you go to that level, you have an intrinsic error of that stochasticity that's involved. But apart from that, at least you can distinguish those relatively clearly. And then here, there should be some that light up. We screened here about 10 to the 7 of them, so only very few sign up. In this video, I think we had to wait for a very long time to find something where we have 2 or 3 events, but they get irradiated here and then show up. And then these electrodes would turn them into a direction that they would not normally take. And then you sort the men from the boys and go through rounds of evolution here. Now, again, there are very few positive hits. So initially, in the first round, we thought we're getting too many false positives. It's all sorting faulty. And the problem is now that we've Poisson distributed. And what you see here as false positives is the very end tail of Poisson distribution that has sometimes multiple cells in it. But by resorting them multiple times, you can actually enrich and make better enzymes. And we've now enhanced both expression as well as catalysis by up to 5 orders of magnitude in these devices in relatively few rounds. Now, we generate droplets at 6 kilohertz, we sort at about 1 kilohertz, and we analyze about 10 to the 7 in 3 hours. And so realistically, that's probably the kind of time that you could spend on it. So all library sizes on the order of 10 to the 7, sometimes a bit more. Okay, and we've applied this sorting system then to metagenomic libraries, not man-made libraries, but libraries that go through the same cycle that I've just shown you, but where the source material comes from the environment. And we use that to get direct to access of biocatalysts that we didn't know before. And I come back to that point later again. We used as bait activities things that seemed useful and difficult. We looked for triesterases that are important for bioremediation and for sulfatases. Both are hydrolytic reactions that we hope would be relatively common. And this is probably the most thermodynamically difficult reaction that's in nature. And we used libraries that we could get from wherever we could get our whole hands on. We borrowed them from different labs and just combined all of them. They come from sludges, ponds, soils, from cow rumen and so forth. And then constructed plasmids that have insert sizes from 1 to 5 kilo base pairs. And we have about 1 million members in total. And that is probably the largest library that ever has been screened with such a functional test, not in vivo, but with a functional test. And then we get hits out. And I don't want to get into the specific identity here, but I want to illustrate where these hits are. So normally, when metagenomics is done, you start off with sequences you think you know, and then you take things that look similar, right? And most of the annotation of proteins that we have is done in this way. Now, if you don't have a working hypothesis, if you don't have an enzyme that works, or if you don't want to find the same enzyme that you've looked before once again, then you have to use this functional metagenomic approach, and then, and that's indeed what we do, you find very different proteins that are not clustered together like a bioinformatic analysis would be, that would look a bit like a phylogeny, but they are unexpected corners of sequence space. And I want to illustrate this with one of the many clusters that we found, these 10 different phosphotriesterases here. This is one that maybe was expected. One of them looks like a triesterase that was already there, but the others are in completely different environments. And here, for example, you have neighbors of that are methyltransferases, deaminases, ureases. Some look chemically similar, but effectively they are in a cluster that hasn't really been fully annotated with functional experiments. That's why we display this as no color. And so these sequence similarity networks tell us that we actually are going further into sequence space than a direct evolution experiment would be able to do. And we find with some of these motifs that were not there. So here, for example, is a cysteine protease that, again, maybe is not unexpected, but there has never been a phosphotriesterase that in the bacterial world that would have this kind of motif that otherwise is very familiar to proteases and other hydrolytic enzymes, for example. Now, if we look at these substrates, we don't even presume that they are triesterases because the environment that we took the DNA from was not exposed to these biohazards. And so we checked whether there are other activities that these particular reactive active sites confer. And indeed, all of them were promiscuous, and they show a large range of activities and seem to be sensitive mainly to the substrate charge. But the common theme there is that we seem to have identified active sites that have so much oomph that they react with anything that requires a nucleophile and is prone to hydrolysis effectively. Okay, so is it possible to do this experiment with other ways? And so we get 8 new triesterases from about 10 to the 6 clones. That would mean that if you are very rich, like Diversa, a deceased company, you'd have access to screening 10 to the 5 with robotic equipment, but you will only get one, so you have to do the experiment many times. And even if you're at home with a relatively bad assay in the lab, you probably wouldn't go beyond 10 to the 4. So that would mean you'd have to repeat the experiment 10 times to find just one of these clones. So metagenomic explorations really require this large sequence space. And also we have a relatively good dynamic range because we can make the droplets in such a uniform way. Okay, so we would like to make that simpler because it was quite complicated to build, in particular, the sorting mechanism. And therefore, we try to think of technologies that are very easy to implement, or at least easier to implement, and have a higher chance of being available to everybody. And here's one. This is when we take the original emulsions and then turn them after they come out here into a different chip with a reversed surface character, and then make double emulsions out of them. Both of them are monodispersed, so we can still do quantitative measurements, but these are now in an aqueous overall environment, and you can take commercially available cell sorting equipment that requires an aqueous phase, like a flow cytometer, to sort them. And we've been able to do this at a relatively high throughput, and we get sort of 100,000-fold enrichment. And that's something where you just have to build the chip, which is relatively straightforward, but the sorting comes from a commercial instrument and therefore becomes much easier. And the second simple technology is one where we immortalize the compartments by co-compartmentalizing agarose and alginate in the gel, together with DNA and substrate and all the other goodies that we had before. But now we let that cool from 37 to 25 degrees and make a gel bead inside, and then add a shell when we quench these droplets and open them up. And the good thing then is that if we use a polyelectrolyte layer here, we have a size selectivity of exit from the compartment. So product, if we label our substrate with a short oligo, we can trap product in there, and then these beads here are a bit larger than normal cells, but they can still be sorted in the flow cytometer. And once you destroy the polyalanine hydrochloride or alanine layer that has formed on the outside here at high pH, you can take the DNA out again, and you can recover the identity of your catalyst by sequencing afterwards. And so we do the same thing. We express, we form droplets, lyse cells, wait for the catalysis to form, and we can wait seriously long there for 6 or 7 days. We've done that. And then the gel shell bead formation allows you to then select based on a fluorescence of sort of a solid entity over here. Okay, now after all that, it's often very difficult for the students to go back to normal pipetting, because no one likes, after having done such high throughput work, to work as we did in the last century or so. And so we've also developed things where we use the ability to make tiny compartments as test tubes to look at more detailed kinetic data that need to be obtained for mechanistic analysis. So we designed droplet-on-demand platforms, and they look like this. We use a microwell here, and then have a very simple pipette that has another pipette inserted in it, and there's a negative pressure coming from here, and then sample is pulled from here, and oil comes from the sort of inner layer that you kind of just see here on the side. And then you're creating monodispersed droplets. Although it's so simple, you're creating monodispersed droplets, and you can regulate what the concentration in the reservoir over here is. And then you get a concentration gradient, and you can use it to get Michaelis-Menten curves or other mechanistic experiments. But maybe that doesn't mean so much to those who are not necessarily biochemists. And I want to illustrate it with painting with the same concentration gradient. We've taken Big Ben, pixelated it, and now created a concentration gradient that reflects the concentrations that are set up over here. This is highly accelerated. Normally it takes about 40 minutes to do that, but we can make concentration dilutions up and down and reflect now a relatively complex sort of mechanistic experiment or painting experiment with that. Okay, so the theme of the conference was whether biomimicry leads us to technology, and as far as technology is concerned, we've now miniaturized assays by 1 millionfold. That makes assays accessible, gives us access to large numbers at low cost. And we can do combinatorial experiments that exceed what we could normally do. The cost of equipment is relatively low, thousands instead of millions, and probably with higher throughput. And we've applied it to direct evolution and metagenomic screening, but there's much more that can be done. For example, single cell experiments are interesting. Dave Weitz has just published transcriptomic analysis with single cells and droplets, and so I think that would be a format that would be maybe as important as the Eppendorf tube in some way. Maybe harking back to the beginning, we've learned something about the process, and in addition to getting just easy access to functional proteins, it would be also interesting to understand better what the imperatives are in an evolution campaign. Normally we do direct evolution, or we've traditionally done direct evolution with a very strangely bottlenecked way. We were so happy that we found one clone because we were screening from small libraries, and then the next one of evolution would be all based on that very clone that we've identified. But that's an enormous bottleneck. Nature doesn't do that. There's much more neutral element in evolution. And so with methods like this, we could widen the range and we could change the selection pressure at each step of direct evolution. We could be tolerant, or we want to be tolerant, at the beginning of an evolution, and then specialize later on. We're now trying to monitor not just one activity, but 3 or 4 activities in promiscuous enzymes, and we may be able to evolve generalists preferentially or specialists preferentially. And at each stage, we can analyze what the entire library represents, and that's effectively a fitness landscape for the protein, and probably there's also mileage in understanding which proteins have promiscuous activities, which proteins adapt very early, which ones just go dead because they're unstable, for example. And I think by doing evolution this way, we can do it faster, but we may also do it better with a better strategy eventually. Okay, so I thank the people who've done the work that I showed here. I showed work by Fabrice Gielen and Balint Kintzis on sorting and measuring in devices. Yolanda Scherli and Martin Fischlechner have developed these gel shell beads. Anastasia Sinchenko and Sean Devenish have worked on the double emulsion that were there, and Pierre-Yves Colline has done the metagenomic selections. We benefited a lot from collaboration initially when we got into this field from people with much more expertise in microfluidics, in particular Josh Edel and Andrew de Mello, and some of the work that I've shown was initially done in collaboration in Cambridge with my colleagues Chris Abel and Willem Huck. So I thank all of those, and I thank you for your attention.